Okay, so I have started recording. The only thing that I talked about pre-recording was a little bit about the Moodle site. We talked about the various review sessions that we may have. You will have a review session before every exam, and you also need to use the Secure Exam Proctor underneath the helpful information. That's a link that takes you to Proctorio to make sure you've got everything installed. Each time you take an exam, you're going to have to click Secure Exam Proctor, even if you've done it before. You don't have to install it every time. By clicking this link, it's priming that Google extension for your browser. So that's how this works. So we're just going ahead and collapse that. Under Start here, you can find a few things. You can find the syllabus, Muddy's Point Forum, and course announcements. Muddy's Point Forum, you guys are already using that, which is wonderful. Let's keep at that. You have the ability of posting things there as well. I will be checking it and communicating, but it's really designed for you guys to communicate with one another. So let's say, Mackenzie's having a question about something. Maria may be able to help her if she posts it to the Muddiest Point Forum. Now, the course announcements is where I'll make major announcements. I don't think you have an option to reply in the announcement chain for everyone, but this is my way of talking to you for mass announcements. You should also get an email here as well. So it's triggered to your email that whenever announcement's posted, you'll get an email to your Surrey account. Make sure that you're using your Surrey email throughout the semester. That's how I will contact you with official announcements. Additionally, another way of contacting me is through Microsoft Teams. Quick show of hands, who's used Teams before? Okay, good, about half of you. So those of you that haven't used Teams, to use Teams, it's not very difficult. You just install it. And one way to install it is just Google Microsoft Teams. But there's a few other ways you can get it. You might even be able to get it from My Nightlife. But make sure you are installing Microsoft Teams and not something bogus. If we just get a quick Google search, Microsoft Teams, it should be this one right here. Free calling, chat, collaboration, that kind of thing. Sign up for free. I think that's how you sign up. I don't know where the download link is. There it is, download Teams. So that's how that works. So for Teams, what we're going to be using with Teams is we're going to be using that for one-on-one discussion. So let's say Courtney had a question, and it was maybe something that email wasn't really set up really well for. She could be like, hey, I'm having some trouble on a speciation. Maybe that's the topic that she wants some help on. She can send me an email or a Teams message, either one. We can sort of set something up to where we have screen shares or video calls or what have you. And it's like a one-on-one synchronous session. But it's nice because it's through the college and it's official, and I like that kind of aspect of it. I have it on my phone as well. So if you were to need to get in touch with me, that's a great way you can get in touch with me. You can send me messages. It's just like texting. You can make calls just like you video called me, or voice calls, either one. It's a pretty handy platform for all those things. And we leverage it quite a bit at the college. I don't have a Teams page set up for you, and we will not be having synchronous sessions for the class-wise through Teams. So all of your synchronous sessions will be done in Collaborate, and the reason that we're doing them in Collaborate is because I like having a one-stop shop. Everything's in Moodle. That's kind of keeps things nice and clean. What we'll be using Teams for is for one-on-one discussions. And those are almost always prompted by an email or a Teams message or something. So does that make sense to everybody? I hope so. That's sort of how we're going to approach communication. So any questions so far about anything?
I have one.
Sure, Isaac, go ahead.
Will we use Proctorio? So you see those quizzes that you have, will we use Proctorio for all those as well?
Good question. No, sir. Proctorio is only for the exam.
Oh, yeah.
So Proctorio will be used for exam 1 and exam 1 only. It'll be used for exam 2, exam 3, exam 4, but nothing else. We don't use Proctorio for any quizzes or other assignments.
Okay, thank you.
Yeah, no problem. That was a great question, and something I'd like to add to that. It's kind of prompted me to something. Those quizzes, I'm sort of expecting you to be using resources to work on these quizzes. Okay, so the module quizzes they're not closed book, anything like that. I'm sort of expecting you to be using resources to answer that. They're also unlimited for a reason. You should take them until you make 100 of them. Here's the idea behind that. If you take the quizzes and you keep taking them and you keep taking them and you keep taking, you make 100 finally, that's studying is what you're doing, in fact. So that's a really, that's something that we use a lot in the class. It's pretty handy. Here's the way that I suggest students take online quizzes for online classes. I suggest you go through and take each quiz by themselves without study. So for instance, if I'm going to work on evolutionary thought and natural selection, I will take that quiz and I won't study at all. I'll just go in and I'll take it. And I'll see how I do. If I make a 30 on it, I go, okay, that's my baseline. So then after doing that, I go in and start studying these books. And stuff may be spread out through all these books. And I'll go through and I'll say, okay, what are some things when I'm looking through these notes, excuse me, I'll kind of show you here. What are some things that maybe pop up to me? Like, okay, I remember talking about theory and what a theory is. Okay, so I'll write that down in my notes. Like, oh yeah, okay, so I probably need some information about a theory. What is a theory? And I'll write down what it is, and I'll try to sort of explain it a little bit to myself and just kind of take notes as I go. Then, after I've read through those books, I'll go and take that quiz again, and I'll see how I do this time. And I may go up to like 70%. So I've jumped 40 points just by doing a little bit of study, which is great. So I go, okay, well, during that quiz, was there anything that, like, man, I had no earthly idea what that was? That came out of completely left field, it seems like. There was some questions on there that I really didn't know how to answer. I'm going to have a piece of paper. Somehow I'm going to take some notes on those questions and the topics of those questions. Again, when I finish that session, I'll go and open those books again, start studying through the notes, looking those things up. You might need to take it again and see how you do. If you don't find the answers that you're looking for, or if you feel that you still don't understand, you can harness the power of the internet using Wikipedia. Wikipedia is a great source for stuff like this. Wikipedia gets a bad rap a lot of times in education with academics. I don't have a problem with Wikipedia. I never really have. In fact, I use it all the time in class. I think it's a wonderful, wonderful thing for us to have. The problem is whenever you start using Wikipedia as a primary source. You see, Wikipedia is not a primary source. It's a great source, but it's not a primary source. So whenever you're writing papers, you want to try to use primary sources. Let's say, for instance, that Anna and Autumn wrote a paper, okay, and they did the research themselves. They collected the data. If I were to read their paper and cite their original paper, that's a primary source, okay? But if someone cites me citing their paper, now I'm a secondary source. You see, they're better than me because that's the original source. So with that being said, Wikipedia is a wonderful thing, but just understand what it is. It's not a primary source to be cited in scientific writing. It's a open source and freely available encyclopedia, and it's very, very handy. And I use Wikipedia all the time. You can just, let's search evolution. Okay, so this is what we're talking about right now. Evolution. This is a wonderful definition. It's a change in heritable characteristics of biological populations over successful generations. You see, it's got different links throughout here, explanations, a lot of information here. Now, obviously, that's going to be a lot to read, and I'm not expecting you to read it all, but there's quite a bit of information there if you were to get hung on something. Now, down at the bottom of the page, of all these Wikipedia pages, there's always a references portion. Here, you could find some actual primary sources. Now, not all of these are primary sources, but some of them are. For instance, this here is a primary source. It's called the Niche Construction Perspective, a critical appraisal. It's in the journal Evolution, and it shows the journal volume and the links and various ways of accessing it. This is a great site. This is a primary source. Looks like Scott Phillips et al. Was who wrote that. So we would cite that. That is a source that we could cite. Some of these other stuff like Evolution Resources, National Academics of Science Archive, that's not a primary source. That's at best a secondary source. So we probably don't want to cite that, even though that's probably right. I mean, National Academy of Sciences, Engineering, and Medicine, that's probably a very esteemed institution. That's still not a primary source. So that just gives you a little bit of ideas on how sources go and how to use Wikipedia primary sources, that kind of thing. So, okay, a little bit of information there. That's how I would expect you to do those quizzes. I would expect you to take them until you make 100. Do you have to? No, you don't have to. But why wouldn't you? It's a guaranteed 100. The more often you take the quizzes, the better off you're going to do on them too. So I recommend taking them as many times as possible. That's a great way to study, because some of the questions on the exam come directly from the quizzes. So that's pretty handy like that. There's also, just to kind of warn you, he's been doing really good tonight, but I do have my little buddy in here. He's just turned 3 a few weeks ago. So he's kind of chilling out watching Blippi right now, but it's hard telling what will happen. There might be some times when he gets pretty disruptive. If that's the problem, if that's the case, I do apologize, but we'll kind of work through that. Me and my wife have had to sort of switch around our work with the pandemic, so she has started working in the afternoons and evenings so that I can teach in the day, and so she's busy right now. But anyway, a little bit of background there. So that being said, I think that's covered most of the administrative things. Let's see. Upcoming assignments, yeah. So we want to talk about what assignments we have coming up. In this section, we have 3 quizzes here: evolution, thought, and natural selection, mechanisms, evolution, and speciation. Those are the 3 quizzes we have for this module. We do have one message board. It is a human evolution forum, so we have it posted up there. I don't think anybody's posted in there yet. Yeah, we've had one post in there so far, so that's good. We have a lab, and through that lab, we're going to have a few different assignments that really you're going to turn in 2 different things for the lab, and I'll be giving you the instructions a little bit later on. Okay, so that's your assignments for this module. Also, we are in the middle of something that's very rare, being its extreme, I don't know, how can I word it? Serious, I guess it's not really a strong enough word. Significant worldly historical event with the pandemic that we're going through right now. I like to let my students ask questions about that as much as possible. It is very scientific, obviously, but I've tried staying caught up on the research as much as possible. I'm not, a virologist or even a human anatomist, so it's kind of tough for me following everything, but I do try to stay caught up on that. If you have questions, I encourage you to ask them. If you have comments, if you saw a really interesting study or paper or anything, we can talk about the vaccines, we can talk about the virus, pandemic, safety protocols, PPE, whatever you want to talk about that's scientific related, we can talk about that in terms of COVID. I want to give everybody the opportunity each time we meet. We're not going to meet every week. We'll probably meet twice per module. We'll have one meeting that addresses the lab, which is what we're going to be talking about here in just a few moments. And then we'll have another meeting, which is a review for the exam. So that's my plan. If additional meetings are needed, we can plan those. But as of right now, we'll be only having 2 synchronous sessions. The synchronous sessions are not required. However, being here is highly recommended. The reason is by being here, you're able to ask questions and participate. One thing I really want to warn you about, don't let this class become a button clicking class. I know that a lot of times for folks that are taking online courses, we think about things and we say, well, I don't want to really take biology, I'll just take it online. Or I really don't want to take English 111, I'll just take it online. That's a problem because online's harder than seated first. It's already harder because you have to teach it to yourself. And then when you're taking these kind of difficult topics online, that really makes it even harder. So how I can increase your chances of success is to have you being engaged as much as possible. I'm trying to prevent this from becoming just a daily task or a chore that we do every few days. I want it to be more than that. And how I can do that is by really increasing the student engagement. I'm not crazy over message boards, would use them some here and there, but for me, that's not my best engagement tool. The best engagement tool that I have is what we're doing right now, these synchronous sessions. So I'll be having these every module. Please, I really do ask that you try your absolute best to attend these. I know that no one wants to be here Thursday night at almost 10 o'clock, but that's what everybody sort of kind of voted for, and these are really a needed, we'll call it a necessary evil, I guess, or needed. Last semester, students loved them. They went really well for the online general biology too last semester. And I think that engagement really increases. And when you have student engagement go up, you're going to have student success. So that's the goal. So, okay, any questions?
Yes, I have one.
Go ahead.
So when I look on the quizzes, it says that there are limited attempts. How many attempts are there?
They should be unlimited. If they're not, I'm going to fix that right now.
Okay. I think it says it in like little writing that there's like limited attempts, but I just wanted to ask and make sure.
Sure. Let me go through here and check this out for us. Yeah, I see that. Yeah, I see that text. I don't understand why it did that either. It should be all the same font. I really don't know why that's the case. Let's see if I can fix it. I think that may have fixed it. Timing, grade. Yep, they that one's unlimited. Let's see here. Let's check this one out here. Yep, grade. Okay, that one's unlimited. So it looks like mechanisms and speciation are both unlimited. Looks like I'm having just a little bit of trouble accessing some of this other information right now. The Moodle site's giving me the finger a little bit. Here we go. Okay. So if this works here, I'll clear that. Timing. Yep. All right. So the ones that's up there are in fact unlimited right now. So make sure that let's make sure that comes through. Okay. Yeah. Okay. So everything's squared up with that. They are unlimited. Throughout the semester, if you ever see a quiz due date that's really strange, like that speciation was like in September. That's just something I didn't change, or I changed it and it didn't stick, which happens more often than I would like. Something else was with these quizzes. If you go in it says you only have 5 attempts, let me know. That's been a mistake, or it's been a mistake on my end. So obviously I need to fix that. So just let me know, shoot me a message or email or what have you. Exams will be only 1 attempt though. So when you do take an exam, it will be closed note 1 attempt. Okay, so what are some other questions we have?
I have a question.
Go right ahead.
So when I go to my nightlife under my middle classes, your class isn't showing up for me.
So that's pretty common for a lot of folks, and I don't know why that's happening. It's being corrected by distance education. Again, this is one of those things that is kind of out of my, I guess, control. But you can access Moodle differently. I'm sure that you probably already have. You can go to online.surrey.edu. And when you do that, that's sort of, we'll call it the back door that allows us to access this information without having to go through my nightlife. And if you ever have trouble with my nightlife and showing your classes and that type of thing, I recommend trying that online.surrey.edu.
Okay, thank you.
Yeah, you're welcome. And anytime that happens as well, you can always send me an email or a Teams message, and we can sort of try to sort that out. Okay. Is there any other questions about anything? All right, if that's the case, then we're going to go ahead and get started with the lab portion of tonight then. So this lab is around evolution, but we're going to sort of look at evolution from a little bit different perspective than perhaps you have in the past. A lot of times people think when they hear evolution, they just sort of they really get hung up on survival of the fittest and that kind of thing. And they let that be all that evolution is. But there's more to it. And one of the things that we have to understand is how we have relationships. So relationships between organisms tell us a lot of information that gives us evolutionary relationships. Let me get a drawing here to sort of show you. So what I'm going to do is I'm going to share a blank whiteboard, and then I'm going to just do some kind of simple drawings here. Let's say we have this tree here. Okay. So here is our tree. We'll say this is A, B, and C. Okay. Now, if just by looking at this, what do you think? Anybody can comment as well. Which two letters are more closely related to each other? A and C or A and B?
A and B.
That's right, A and B. That's correct. A and B. And that's very obvious with this image. You can see that very clearly right here. A is more closely related to B because it has this sort of grouping here. But when we put a timeline up here and we say 0 and now, and we change A to something like, we'll just say, I'm trying to think of a creature that's A and B. Let's say ape, bonobo, and cat. Now this is not perfect, so don't really look into the evolutionary relationships here. But what we can see is we can say that ape and a bonobo are more closely related because they share a more recent common ancestor. The circle and the arrow is the common ancestor that an ape and bonobo share. The second circle that I've just drawn is the common ancestor that an ape, a bonobo, and cat shares. By dropping a vertical from those points and looking along the timeline, you get a really nice idea of when that common ancestry occurred. You see, you have a more recent common ancestor between the ape and bonobo than you do between the ape and bonobo, or between the ape and cat, or the ape, bonobo, and the cat. Does that make sense? Hopefully that should sort of explain some stuff fairly well on how trees work. Okay, let's go ahead and draw another tree then. Let's see, would anybody like to volunteer some family information? What you need to have is you need to have a brother and or sister and a first cousin. Does anyone have that combination that'd like to share? Okay, Elena, go right ahead. So see your hand there up first. So let's see here, we'll write her name down. So Elena, and you have a brother or sister?
Do they have to be full blood siblings?
They, let's see. No, they don't have to. No, they don't.
Okay.
So we're okay. You have a, is it a brother or sister?
I have 11 brothers and 9 sisters.
Oh, goodness. Wow. Yeah, you got a bunch.
So take your pick.
Okay. Yeah. Just pick one of them. It doesn't matter who.
Christopher.
You can just Christopher. Okay. Okay, so Christopher. Okay. And this is brother. Okay. Now, you and Christopher share at least one parent, is that correct?
Yes, we share a father.
Okay, share a father. And do you have a cousin on your father's side, a first cousin that you and Christopher have in common?
Yes.
Okay, and what is that cousin's name?
Ryan.
Okay, Ryan. Okay, so what we're going to do is we're going to make a quick phylogenetic tree now of just the information that we've been given. Okay, now, obviously, Elena has quite a few more brothers and sisters, and she probably even has more cousins. But with the information presented here, this is all we need, so we should be okay. Now, let me just ask you some questions here. Who is at the purple circle? Who is that?
My father.
That's right. It's Elena's father. Now, who's at the orange circle?
His father.
Yeah, his father or mother, right? Either way. As if Ryan, Christopher, and Elena all share the same set of grandparents, then it could be either one right here. But you see, the common ancestor is another generation back for Ryan than Christopher. So if we're comparing how closely related Elena is to Christopher versus Ryan, then we can see using common ancestry that she's more closely related to the father than the grandfather or grandmother. Okay? Or more closely related to her brother Christopher because they share a common ancestor that's more recent than does Ryan. The common ancestor with Ryan is additional generations back. It's either the grandmother or grandfather. Whereas with Christopher, it's her father alone. Does that make sense? Hopefully everybody sort of gets that a little bit now. So that's how you draw a phylogenetic tree. Phylogenetic trees are something we're going to use quite a bit in this class, specifically with this section, animals, and some in plants as well. But they're a very important thing that help us look at the relationships amongst different creatures. So we're going to try something here real quick. Since this is sort of a lab kind of a scenario, what I'm going to do is I'm going to post up some images here, or excuse me, not images. I'm going to post up some names. And then I'm going to allow you guys to answer some questions. So here are the names. I'm just going to copy them and paste them here. I've posted that in the messages. So those are the names. And what I'd like for you to do is I'd like for you to Google search those. Now I'm going to demonstrate one or two of them for you here. So we'll just do the first one here. We'll do Micropteryx dolomew. So I'm going to go and do a screen share for us. Okay, so we'll do a quick Google search of this animal. We'll look under images. I want to look at an actual picture, not a drawing. This is a pretty good picture right here. You see, this is a nice image here that shows us. This is also another good image. You can see this is the smallmouth bass or Micropteryx dolomieu. You can sort of see how this animal looks. I'm going to now look up the other. I'm going to look up an additional one here for you. So let's do Micropteryx salmoides next. So Micropteryx salmoides is the largemouth bass. So let's just search it. Under images here. And let's see. Here we are. Any of these will work. This one's pretty good right there. So there's Micropteryx salmoides, the largemouth bass, and there's Micropteryx dolomieu, the smallmouth bass. Okay, so then we got to have one more to sort of start this process. We're going to look up Micropteryx punctulatus. So we'll just do, again, a Google search for Micropteryx punctulatus. This is the spotted bass. And you can see, here's the spotted bass right here. It's even ncfishes.com. So now what you need to do is you need to look at these three, dolomieu, salmoides, and punctulatus, and you think about how can you group those? Are these in three distinctive groups, or should I start grouping them into two groups? Maybe if I had to force you to put them in two groups, what would you do? Would you do probably salmoides and punctulatus in one group and then dolomieu? Or would you do punctulatus and dolomieu? How would you organize them? What would you do? Anybody want to comment on that? Well, that's okay. Okay, let's see. Yep, salmoides and dolomieu. Yeah, that's a pretty good way of doing it. Not bad. You know, there's quite a few different ways you can organize these things. What I'm kind of interested in you to do right now is just to Google each one of these names that I've pasted up here and go into images, try to find a nice image photograph of them, and then start grouping them. And if you want to, you can just do that on a scrap piece of paper in some notes somewhere. And just sort of see if you can make two, no more than three lists, okay, of these animals. So try to keep them in two, no more than three lists. Does everybody understand what you're doing? Just by purely how they look, just sort of how these animals appear, we're going to base our organization off of that. So just sort of group them based off how they look. All right, so I'm going to give everybody just a few minutes to do that. All right, guys, I'm back. I'll give you guys a few minutes to organize yourself there, kind of see how everything fit together. Would anybody like to share any of their lists with us? We can type it in. That'd probably be the best way, or you can log on and speak it if you'd like. I'm just curious on what your thoughts are just based off of how they look. All right, let's see what we got here. Dolomieu, punctulatus, cosi, cataracta, and notitis together, then Samoyeds, Florida, and Trusuli together. I could see that. I think that's a pretty good way of doing it. I forget what Trusuli looks like. I always got to search for that. Yes, the Guadalupe bass. That's right. Yeah. Yeah, I think that's a pretty factual way of doing it. In fact, it really doesn't matter how you do it. What you're doing is you're simply trying to get some way of these animals looking like one another in groups. And what you're going to do then is you're going to then do something like this. You're going to draw a tree based off of those. So let's say we were to use, let me drag some information over here so I can see. Okay, let's say we're going to use Kaylee's information. What we can do is then we can say, well, amongst that group, Dolomieu, punctulatus, Cusa, cataracta, and notitis, wonder how they really looked with one another. Well, we'll say, I think punctulatus sort of looked like its own little dude, okay? Okay, we'll just say it's a here. But then, up next, I kind of think that Dolomieu has its own little look here as well. Then we'll say, well, I think then Cusa down here. But then I had finally cataracta and notitis. Now this is pretty poorly written. I know I'm trying to write it with this quick little pen thing here. I'm spelling it wrong too. Notitis. This would be US there. Then we can drop this down and we can have the others over here. Let's say you had something like, I don't know, Salmoides. And then you had Floranus. And then Trusuli. You see, I've built a phylogenetic tree here based off of how I think they look. Now, it could change. For instance, if you thought, I don't know, Dolomieu maybe look more like notitis, you know, it would go here. So you can switch any of these around. However, you can also change the organization. Let's say you know, maybe you thought these were just in two groups instead of broken down as a ladder like that. There's many different ways that you can organize this. So don't be afraid of using different methodologies and stuff. But you need to stop that. You need to stop. You need to stop, okay? You gotta stop. You gotta stop. Okay. So that's sort of how I want you to draw a tree. And you're going to upload that under Black Bass's tree. So that's the assignment that you'll upload it under. And it says upload a photograph of your tree here. The reason I said that is that most folks is probably going to want to draw them out by hand. If you want to draw them out on a computer, by all means, go right ahead. You can make an electronic copy. But I figured most of you probably want to draw them out by hand because it's a little easier drawing the trees when you have pencil and paper. But I do want you to use this structure where you have these sharp angles up and to the side. Sometimes students like to draw phylogenetic trees, or you may hear them called cladograms. Cladograms and a phylogenetic tree are very similar to one another, and they can both really sort of show you the same thing. But I'm going to show you a quick image here from Google Images. So let me just share my screen too real quick. So you see, if you just search cladogram, you have two different ways you can do it. You can do this straight line like this, or these broken lines, as sort of these angles. I much prefer the right-hand side over the left-hand side. I think it's easier to see evolutionary relationships. So please, you try to use this. If you need a little quick explanation on how to draw them, you can always search for cladogram or phylogenetic tree in Google and just go to images, and you can see how to draw them pretty easily. That A, B, and C, that's your base cladogram. So if you ever forget, it's a good way to remember. Just think about how A, B, and C are related to one another. And that should get you somewhere. So what you're going to do is you're going to draw that tree, and then you're going to upload it under Black Bass's tree. Buddy, come here. Come here. Okay, I need that, okay? Okay, thanks. So then after you've got that uploaded, then you have another assignment you need to do. So after you have your tree uploaded, you're going to then look at this tree here. Stop. Okay, so this tree is called Black Bass's phylogeny collapsed. Now, hey guys, my apologies there. I think I got it taken care of finally now, and I've went ahead and updated the link for you. For some reason, I had a hard time getting this accessed. I had to save it as a TIFF and then as a JPEG and then convert it over. But anyway, my apologies. It is now fixed. I don't know why that was that way. I never had any problems last semester. But it is fixed now, and I do have it uploaded. So, and I'm screen sharing here with you. It should be popping through any moment. There we are. So what we're looking at here is a phylogenetic tree of the animals that I showed you a while ago, plus one more. I added Lepomis microchirus in there. That's, excuse me, that's a bluegill. I just had to put that in there so I could organize these animals a little bit better. But what this tree is this is a tree based off of a very specific gene called the CO1 gene. Or this could be even cytochrome B. I can't remember, but it's a very specific gene. I think it's the cytochrome B gene. And what happened is, excuse me, some researchers went and collected each one of these fishes. I know fishes sounds funny, but it's actually correct. Fishes is. So a researcher went and collected each one of these fishes, and by collecting them, took samples from them. Either they sacrificed them, or they were able to take fin clips, or somehow they were able to access the DNA that these animals had. Once you're able to access the DNA, then you're able to do a lot with them in terms of comparing them. And specifically, we're looking at a very specific portion in the DNA called the mitochondrial DNA. You have two sources of DNA in your body. You have a mitochondrial DNA, and you also have nuclear DNA. Mitochondrial DNA is DNA that comes from your mother only. And it's found only in your mitochondria and within your cell. The gene that we're looking at on the mitochondrial genome, which is all of the DNA, is the cytochrome B gene. That cytochrome B gene is involved within the electron transport chain portion of cellular respiration. If you remember the cytochromes at the end, it's a gene that helps create those proteins. But we're not going to get hung up on what the gene does. We're more interested currently in what we can use the gene for. Researchers have taken these genes, and remember, genes are just A's, G's, C's, and T's, and they've arranged them and they've organized them such that the genes from each one of these animals is lined up as much as possible. So for instance, if you have a column with all A's, they'll have A's in there, and then B's, or A's and G's and C's and T's and so on and so forth. And you're going to have gaps. Sometimes some animals may have a C there, and some animals may have an A there. But by moving them around and put these gaps and spreading out, you get what's called an alignment. And an alignment then gives us the ability of building a tree, just as we see here. As we see here, we're using the cytochrome B gene. You see punctulatus is grouping very tightly with dolomu. There's actually only two groups here. And then we have Cusa, Cataractae, Trasuli, Natitis, Salmoides, and Floridanus in their own groups. So you almost have this grouping here. Now again, we're going to ignore Lipomus macrocyrus because that's just our out group. We had to include that. That's a bluegill. But we're going to focus on these others. And the others we're going to focus on specifically is this grouping up here and this grouping right here. And if you notice, the branch length is very, very short between this. That means these animals are very, very close together. Whereas if you can see, the branch length between Salmoides and Floridanus is much longer, which states that these animals are much more dissimilar than these animals are here. But when we were looking at the actual photographs, if you look at the photograph of Salmoides and Floridanus, it looks almost identical. You can't hardly tell them apart. Whereas Punctulatus and Dolomu, you can almost always tell those apart. So what we're getting at here is the idea of using morphology, or how something looks, versus using the genetics. You see, when we're using genes or the entire genome, and we're using that to help us to determine how something's evolutionary related, we generally get a much higher level of precision and even magnification than you do if you're using pure morphologies alone. Since the genetic revolution in the 90s and through the 2000s, we're able to do things like this that we were never able to do before. Used to, we just simply had to look at, see how things looked. Okay, well, that has black scales and this one has white scales, so they must be slightly different. Whereas we've learned that color is a very poor indicator of how something looks different, and so is shapes. These things can change pretty dramatically. But how does this tree and the organization of this tree compare to what you have? Is it very similar or is it very different? These are the kind of questions you want to start asking yourself. And this really starts getting at some really important things, like what even is a species? You know, a species we define as a group of individuals which live in a similar habitat and can reproduce and produce fertile offspring. That's what a species is. That's the biological species concept. Now, there's a few other species concepts, but the biological species concept states that two individuals that can reproduce and produce fertile offspring are said to be of the same species. So, can Dolomu and Punctulatus reproduce and produce fertile offspring? You know, they actually can. You can have hybrids, smallmouth spotted bass hybrids. You can also have smallmouth Cusa hybrids. You can have smallmouth, largemouth hybrids even. You can have all these different hybridizations throughout these. So it really starts challenging our idea of what a species even is and if a species even exists in the first place. But these phylogenetic trees really help us do that. So the point of this assignment is for you to look at your tree and then look at this tree that I've just uploaded and compare and contrast the two. Then you're going to start getting at the idea of, well, why does your tree look so much different? When we look at, let me just ask you a quick question here. Let's just do Micropteryx, Punctulatus, Micropteryx. Let's do here Cataractae, and then let's do Micropteryx. We'll just do Salmoides. Okay, so I'm just going to kind of put these up over here so you can see them. I think, let's see what's going on here. Okay, so should be one more. Here we go. Okay. So here's the three we're going to focus on here, just kind of real quickly. Let's just look at some pictures of them. So here's Punctulatus. So here it is. There's one picture of it. Here's another image. Here's another image. There's another. That's a drawing, but there's a nice picture. Let's look at that one. Okay. And then let's compare that one to, say, I don't know, what about this one here? That's pretty good. And then let's look at Cataractae and compare it to this right here. And in fact, let's put one more up there. Let's do Micropteryx Dolomieu, which is smallmouth basses. Now, I'm going to make an assumption that maybe I'm wrong, but just by the way I'm looking at things, when I look here, I see Micropteryx punctulatus here. I see it as being more similar in how it looks to largemouth bass, which is Salmoides, than it does to Cataractae or Dolomieu. Do you agree with that or disagree with that? Do y'all agree with what I'm saying here? I hope so. All I'm saying is I think these two fish look similar and these two fishes look similar. Now, if we look at the tree tells a different story. So let's look at the tree here again. Sorry, I've got so many tabs open, it's hard to keep track of everything. I'm trying to get it all on one nice page here for us. So if we look at the tree, you can see punctulatus, they're saying is more similar to Dolomieu. And then Cataractae maybe even more similar to Salmoides. But if we look at just how they look, that's not the case. Look at how this animal looks. Do you see? They look more similar to one another with their colorization and color patterns. Dolomieu and Cataractae, they almost look identical except for just a little bit more cryptic colorization. But if we look at this and this, oh wow, that's even tougher to tell apart. Maybe one's a little lighter and darker, but they're very, very similar. What we're looking at is you're looking at something called an ecomorph. Does anybody know what an ecomorph is? Maybe want to take a stab at maybe what an ecomorph is? I know it's a question that you may not have heard of before. So when you hear morph, what comes to mind when you hear the term morph?
Changing into something else.
Yeah, something changing into something else. I like that, yeah. And by changing, you're generally probably focusing on how something looks. And that's what we mean by morph, is if you hear the term morphology or the term morph specifically, what morph means is how something looks. So if you were to hear us say morphology, that's the study of how animals look. Now that's an important thing, but it's limited because when we look at these animals here, they look similar, but they're actually differently related. Looking at their genetics helps us look more at the evolutionary relationships, because that means that punctulatus and Dolomieu share similar ancestry, whereas that ancestry is much closer than it would be to, say, Salmoides, even though they look similar. More than likely, the reason they look the way they do is due to their habitat. And I can comment specifically on these. Punctulatus and Salmoides tend to live more in lakes than they do rivers. Now, we do have quite a few of them in rivers. My apologies. So the lakes and rivers are generally bigger, deeper, slower. In these cases, if it's bigger and deeper and slower, you need to be a little bit lighter color actually to blend in, because then as you go deeper, if you have these darker colors, you're going to stand out more in a dark background. Whereas if you live in shallow water and you're really light, you'll stand out really bad. So being dark helps you blend in a little bit better. So see how this smallmouth bass has almost that camouflage, that cryptic colorization? Well, you also have this with the shoal bass or the Cataractae. And these animals both, they have that cryptic colorization because it's camouflage, really. They live in shallower water. When you live in shallower water, then you're able to blend in a little bit better with that smaller background or that shallower background. It may be darker, and you might be able to blend in a little bit better than you would if it was lighter and deeper in a lake. Does everybody sort of follow me a little bit there? What we're talking about, again, is an ecomorph, and we're talking about convergent evolution. That's another term. Convergent evolution is body shapes or forms that animals tend to converge on or come to without actually ever evolving that way. A good example would be the wings of a butterfly, the wings of a bird, and the wings of a bat. There's no common winged ancestor that those animals all share in common, but they all do fly because that's convergent evolution. That's evolution converging on a specific body shape or appendage or trait that allows the animal to have a specific characteristic like flight. Wings work very well for flight, so we have flight. Another good example of convergent evolution is eyes. There's many animals that have eyes, but there's really no common eyed ancestor. For instance, the eyes of a human, the eyes of a rabbit, the eyes of a snake, the eyes of a spider, and then the eyes of an octopus. There is no common eyed ancestor there. Now, yeah, there's a common eyed ancestor amongst vertebrates like rabbits and squirrels and humans, but you keep going back, you'll eventually find that eyes or vision has evolved quite a few times independent of one another, and that's convergent evolution. So what we're looking at here is we're looking at body shapes or morphs that's being converged on due to habitat. Shallower habitat means you probably need to be darker in color and more cryptic in colorization. Whereas in deeper habitat, it's actually better to be a little bit lighter in colorization because you don't need to spend all that time making pigments and stuff just to stick out whenever you're in deeper water.